Hi everyone, and thank you for dropping by. My name is Anna Vlot, and in this video I present to you my poster on SEMITONES, which is a tool for single-cell marker identification by enrichment scoring. So now first for a little bit of background. So the identification of markers of cell identity is a common step in single-cell omics analyses, and is most commonly done by differential testing between clusters of cells with the same identity. However, obtaining these clusters of cells with the same identity is non-trivial, in particular for developmental or differentiation data, where cells gradually change along a trajectory and cannot be discretized into cell types or states in a meaningful way. So to circumvent this clustering step, we introduce a clustering-independent method for de novo marker identification from single-cell omics data, called SEMITONES. So the workflow of SEMITONES is as follows. First, we select a set of cells that is representative of the full sample population, called reference cells, which I indicate here in pastel green. Then if I take this dark green reference cell and assume that we are looking at single-cell RNA-seq data, we define a marker gene as a gene that is only expressed in the reference cell neighborhood, as shown in orange, and in contrast, uninformative genes are genes that are non-selectively expressed, as shown in purple. From this it follows that there is a linear relationship between the similarity to the reference cell and the gene expression vectors of marker genes. If we then use simple linear regression to obtain the beta coefficient that quantifies the strength of this linear relationship, this coefficient can be interpreted as an enrichment score that indicates how much more likely it is that we find high gene expression values at the top of a list of cells ranked by their similarity to the reference cell than at the bottom of said list. Using this approach, we get high scores for genes that are only expressed in cells similar to the reference cell, as shown in orange, and low scores for genes that are non-selectively expressed, as shown in purple. Then next, SEMITONES can test for significance using a null distribution of scores for permuted gene expression vectors. In practice, this null distribution is a distribution of enrichment scores for randomly expressed genes. Significance is then declared at a certain number of standard deviations away from the mean of this null distribution. Then lastly, SEMITONES can construct co-enrichment graphs to infer regulatory interactions. This is done by computing enrichment scores for sets of genes and constructing graphs where nodes are features and the edge between each of these features is weighted by the enrichment score of those two features. So let me now show you a few applications of SEMITONES. So in this first panel I show SEMITONES for the identification of marker genes using single-cell RNA-seq data of healthy human hematopoiesis from the Greenleaf lab that was published at the end of 2019. As illustrated in panel A, SEMITONES identifies overlapping and unique marker genes along the developmental trajectory of B cells, allowing for a highly specific annotation of cell states along this continuum. Also, as shown in panel B, given such markers for all reference cells, we can annotate these reference cells by their cell state. These cell states correspond broadly with the published cell state annotations, which were informed by cell surface marker proteins and therefore served as a valid ground truth. Additionally, we could annotate more specific T-cell and monocyte subsets based on SEMITONES markers. Then here I show that SEMITONES can also be used as an alternative to highly variable gene selection during single-cell RNA-seq processing. So in panel A I show a 2D UMAP of the same single-cell RNA-seq data using only the top 1000 most enriched genes. And in panel B I show a 2D UMAP of the same data using the top 1000 most variable genes. Here we can see that cell states remain more separated in panel A than B, suggesting that genes selected by SEMITONES account for more of the biological diversity than the highly variable genes. In concordance, certain well-known markers show more localized expression in panel A than B. This application is of particular interest when selecting a subset of highly informative genes for targeted sequencing. Then now for an example of co-enrichment graph construction by SEMITONES. So the following panel shows an example of a co-enrichment graph inferred by SEMITONES for red blood cells. To illustrate the biological relevance, I point to connections inferred between KLF1, HBA1, HBA2, and AHSP. For these genes and their protein products, known regulatory interactions are documented. Namely, the genes coding for the hemoglobin proteins HBA1 and HBA2 are known targets of the transcription factor KLF1, and the hemoglobin stabilizing protein AHSP is a chaperone of HBA1 and HBA2. These results illustrate how co-enrichment graphs can reveal putative regulatory interactions. Then finally I want to show that SEMITONES can also be applied to other data types like single-cell attack-seq data. Here I show an application of SEMITONES to single-cell attack-seq data of human hematopoiesis from the same publication by the Greenleaf lab. For single-cell attack-seq data, SEMITONES identifies genomic regions that are selectively accessible in a certain reference cell neighborhood. Since these genomic regions are not generally annotated in a cell type specific manner, We pass the significant regions to grade to get GO-term enrichments for close-by genes, and then annotate our reference cells based on these GO-term enrichments. Like before, most of the annotations overlap with those in the original data publication, confirming that the inferred regions contain cell state specific information. Furthermore, when assessing transcription factor binding motif enrichment in significantly enriched regions, we find biologically meaningful patterns. In transitional B cells, for example, motif enrichment is found for E2A, HEB, TCF12, and AP4, which are known regulators of B cell development. These results illustrate that SEMITONES identifies relevant, cell-state-specific cis-regulatory regions from single-cell ATAC-seq data. So, to conclude, we introduced SEMITONES as a method for de novo feature identification in single-cell RNA-seq and single-cell ATAC-seq data. We suggest that SEMITONES is a more sensitive alternative to highly variable gene selection in single-cell RNA-seq analyses. And we illustrate that SEMITONES can be used for the construction of co-enrichment networks that reveal regulators of cell identity. Now, if you would like to know more about SEMITONES, you can read the preprint on bioRxiv, or alternatively, you can visit the GitHub page where I provide the source code, some small use case examples, and the documentation for SEMITONES. And of course, if you have any more questions, please get in touch via email or social media. And with that, I'd like to thank you for your attention.